So good day, everybody, and also from my side, welcome to this webinar. So I want to give a little bit of an introduction to Revio and a couple of new tools that we are having for data analysis. Let me see if yes, there we go. So as an appetizer before our main speaker, I want to talk with you about our newest sequencing platform, Revio. So with our new flagship, you can sequence 1,300 human genomes per year thanks to its new 4-stage sequencing design. This design allows for parallel sequencing of 4 Revio smart cells regardless of the applications. And each smart cell can sequence a human genome with 30-fold coverage, where 90% of the bases have an accuracy greater than Q30, and that all within 24 hours. This includes the detection of 5-methylcytosine in a CpG context without having to make any modification to the sample or library. This just adds another layer of information beyond the regular 4 bases for a better understanding of the epigenome. Now, I'm sure many of you are already familiar with the concept behind HiFi sequencing. In a nutshell, the technology allows for sequencing single circular molecules, tens of kb in size, multiple times. Basically, the more passes, the higher the accuracy of the HiFi reads, as random sequencing mistakes are identified and polished in the final consensus read. This final read is what we call a HiFi read, and a typical HiFi dataset has a mean read accuracy of 99.9% or Q30. So our HiFi reads are both long and accurate, almost free of systematic errors. And because of the great single molecule accuracy, with additional coverage, you easily achieve very high consensus accuracy of Q50. Because our platforms sequence single molecule DNA with no additional amplification, we see the least bias towards GC contact and sequence complexity compared to existing sequencing platforms today. HiFi reads is also showing its strength in the different research fields. For example, when it comes to rare inherent disease, a lot of cases still are unexplained because short read whole genome sequencing is not able to show us the full picture due to the low coverage in difficult regions of the genome. And they also lack the power to detect many of the large structural variants. The same accounts for many of these other fields that you see here on this slide. The long, accurate HiFi reads do allow you to span repeat structures, discriminate between homologous genes, look at complex structural variants, and provide better haplotyping for greater population resolution. So sequencing data is nothing without the correct tools that utilize long-read data to its fullest potential. We have a suite of tools to analyze HiFi data, which allows for more discovery power than any competing technology. So for small indels, we have deep variant. For structural variant calling, we have PBSV. For tandem repeats, we have a new tool called Target, more about that later. For segmental duplication, we have Paraphase, which can specifically look at genes in segmental duplication, and we're building out this list. For phasing, we are switching now to HiFi instead of WhatsApp, because HiFi also makes use of structural variants. For copy number variations, we have a new tool called HiFi CNV, which basically allows you to replace microarrays or validate previous microarray results. And we are further optimizing our tools for methylation calling. More on some of them later, but as a little teaser, to make your life easier, we are working on combining the tools in one easy workflow that you can run from your own high-performance cluster or in the cloud. So keep an eye on our blog for more information to come. Now, as an example highlighted here are 2 papers from this year showing that HiFi whole genome sequencing detects more de novo mutations. In short, in both cases, they find more de novo variants for all variant types compared to short read whole genome sequencing with the least number of false positives. Important to mention here is that it also depends which reference is used for comparison. The more complete the reference, like the T to T reference, the more variants are found in regions otherwise not explored. And so continuing further on structural variants, it is well known that structural variants, although lower in number compared to SNVs and small indels, they do cover much larger regions of the human genome, as is shown in this figure. If we look in ClinVar, 50% of the structural variants are accounted as being pathogenic. We also know that the number of known structural variants is underrepresented in short-read whole genome sequencing because they were never detected, which will have an impact for detection of diseases caused by structural variants, putting some research subjects through a long medical odyssey, as was the case for this 12-year-old female. Only with PacBio was a 12 kb inversion found, explaining her case. And so as we are discovering more and more variants with long-read sequencing, we know that we need more ways to frequency filter the many novel variants that are found by HiFi to further increase the utility of long reads for researchers. So to address this, we have formed the consortium for long reads. The goal of COLORS is to create a frequency database to help distinguish common from rare variants that may be disease-causing. Think of this as a genome AD for structural variants from long reads. So this slide highlights one of the two I mentioned earlier. Now, to fully utilize long reads to interrogate tandem repeats, we have developed a new software that is designed to provide the best-in-class repeat expansion caller, which is named Tandem Repeat Genotyper, or in short, TARGET. This tool will enable targeted analysis of SDRs, VNTRs, visualization of the results, genome-wide tandem repeat cataloging, discovery of de novo SDRs and VNTRs, methylation calling, and haplotype resolved repeat expansion calls. So the caller uses aligned HiFi reads and is fed the information of the repeat expansion in question. It will show the repeat sequences found in the sample, the repeat length, the methylation status, and more. This can then be visualized using the tandem repeat visualizer. As you can see, this information is provided for both haplotypes. Another tool that was on the list is called Paraphase, which is specifically designed to resolve segmental duplications. So segmental duplications are important because they comprise 7% of the human genome. Many clinical relevant genes fall into segmental duplications. High sequence similarity of segmental duplications poses challenges to read alignment and variant calling. And segmental duplications are hotspots for structural variants, including deletions, duplication, and inversions. And so to show you how HiFi can resolve genes in segmental duplications, let's use SMA as an example. As you may know, SMA is an autosomal neurodegenerative disorder and a leading cause of infant death. It is caused by a lack of functional SM1 gene, and the copy number of the paralog SMN2 determines the disease severity. Now, accurate genotyping of SMN1 and SMN2 is very challenging because these genes are long, 30 kb each, and more than 99.9% similar in their exons. So the ability of Paraphase is to detect the SNVs in HiFi data at SMN1, SMN2. It phases complete haplotypes, it determines the copy numbers, makes phase variant call, and is also able to accurately detect silent carriers. And so underneath is an example of multiple copies of the SMN1 gene and SMN2, where in the third haplotype you can actually see a deletion from exon 7 and 8. Now, as I mentioned in the beginning, HiFi sequencing also allows for looking at the 5-methyl cytosine without additional modifications during library prep. This fifth base information holds potential for better understanding of the epigenome in a phased manner. A quick example can be seen on the right figure. There you can clearly see the two different haplotypes of a DMPK gene, with the top having a large repeat expansion as shown by the insertion and its associated hypermethylated region next to it. Now, some of these mentioned tools also work for more targeted approaches if whole genome sequencing is not desired. You could either use amplicons for easy and cost-effective enrichment of single genes or small regions, or you go for targeting larger regions using targeted enrichment panels from Twist. For both methods, we have great examples, protocols, example data sets, and other resources on our website, so be sure to check those out if you're interested. And as a final mention, we are striving to bring HiFi sequencing and its use to perfection, but some things we can't do alone. And so outside of our core developments, we are creating an ecosystem around us to help improve our workflows on both the front end in the lab, but also at the end for better data processing. One such example in the lab is partnering with different providers for automating sample and library prep, like Hamilton, Tecan, and Thermo. And for analysis with tech companies like Google, who have developed deep consensus that actually runs on the Revio, but also deep variant for improved variant calling of SNVs and small indels. Again, keep an eye on our blogs and don't miss out on any important developments. And with that, I want to hand it over to Adam Ameur from Uppsala SciLifeLab to tell you more about the experience with the Revio system and new applications.
Okay, so I hope that everyone can see me now and hear me. I'm very happy to be here today and talk about some of our experiences of HiFi sequencing and of the Revio system. And today I'm going to talk how we are using this high quality long read sequencing to study human genomes. And we're doing it for individuals, for populations, and even now we're starting to look into the genomes of single cells. I work at the National Genomics Infrastructure in Sweden, and we are a core facility for sequencing. And I should mention that we're not only sequencing humans, but we're sequencing everything. It could be DNA, RNA, and we take on projects from around Sweden and also from around the world. And we have access to both short read and long read technologies. And when it comes to PacBio sequencing, this is something that we've been doing for quite some time. So to the left here, you can see the first installation of the PacBio RS2. So that was quite a big instrument that we got into the lab 10 years ago. And more recently, we are very happy to also have the newest equipment from PacBio, the Revio system. And we are excited to use the Revio system for studying human genomes. And that was one of the first things that we actually tried and wanted to evaluate the system on. And we see the potential here to detect all types of variants. It could be SNVs, structural variants, and tandem repeats. We can do haplotype phasing, look at methylation, and even do de novo assembly. And the aim of much of this would be to try to resolve disease-causing regions and try to understand the molecular mechanisms behind disease. And we wanted also to do this on real clinical samples. As you might know, companies often try and sort of show slides how things look on cell lines, but it can be quite different on real DNA. So that was something that we wanted to do, and also just run one smart cell per individual and see what we can get. So we set up this pilot project to study rare disease patients on the Revio system. And this is a collaboration with something called Genomics Medicine Sweden, which is an initiative to get genomics technologies into healthcare. So there are different nodes within the Genomics Medicine Sweden program at the different university hospitals that are using technologies, and GMS is one way to try to collaborate between these different nodes. So for this particular project, we were interested to study about 15 to 20 clinical cases from different hospitals around the country. And the DNA, as I mentioned, it was extracted by regular methods used at the hospitals, so not these fancy high molecular weight DNA extractions. And for the samples that we looked at initially, there were complex structural variants suspected, so we knew approximately which regions to look at. and there was also other genomics data available, such as short read data and arrays. Okay, and what we have done so far is to sequence 14 individuals on the Revio system, and here you can see the statistics, the number of reads that we get from each of the samples, and the total yield. So we get somewhere between 60 gigabases up to over 100 gigabases per individual, and even 60 gigabases is more than 20x coverage. So I think that's pretty good. And if we look here to the right, we can also see the read lengths, and they can differ quite a bit between samples. And what we noticed was that sort of the best performing samples are the ones, some of these samples were actually extracted using high molecular weight DNA methods, and they were also site selected on gel before doing the sequencing. And I think that's the sort of big difference here. If you do a site selection to get rid of the shortest fragments, you can get more data from a SMRT cell. We also developed a pipeline for analyzing the Revio data. So we start here from the HiFi reads. They are aligned using Minimap2. Then we do variant calling, looking at tandem repeats with the target program. We do SNP calling with deep variant, and we call structural variants with Sniffles2. And after the variant calling step, there is a filtering step that is still sort of under development. But here we're making use of both sort of the other Revio samples that we have been running, and also we have collected data from the human pan-genome reference consortium to do the filtering. And if we look at the results for variant calling, this is a quite big table, but you can see the numbers here of SNVs that we find with deep variant. And if we look at the bottom line, you can see the average value because it's kind of similar between samples. So we have something like 4.4 million SNPs, and then we have 400,000 small insertions and deletions. And we have been doing some initial comparisons also with Illumina short read sequencing. And we can see that about 96% of the SNVs were detected also with Illumina. And to the right, you can see the structural variants. So those are more than 50 base pairs variants that are detected by Sniffles2. And there is about 13,000 large insertions and 9,500 large deletions per sample. We also did some filtering of SVs, as I mentioned, where we are then using the human pan-genome samples, the other Revio samples, and sort of the sample that we're interested to find unique variants. And if we do a Venn diagram like this for one of the samples, you can see that we have 201 SVs that are unique for this particular sample. And if you would like to study, sort of try to find a disease-causing structural variant de novo, this is the variants that you would start by looking at. And I don't think 200 is perhaps a too big number. I mean, if we also restrict it to the ones that are overlapping with a gene, maybe sort of we can get down to a reasonable list in this way. Tandem repeats were mentioned before, and this is just a slide showing how it can look for one of the sample. This is the HTT repeat that is causative for Huntington's disease. And you can see there's a difference between the haplotypes here in the CAG repeat that is slightly longer in one of the haplotypes. And I should mention here that this is not a Huntington's patient, so it's like normal variation that we're looking at here. And we can also then overlay this information with the methylation that can, so we can in this way study the differences in methylation between the haplotypes, which I think is very interesting. We also have another example of the methylation. This is just a known imprinted region where we also can see this difference between the haplotypes, just as expected, that one of the haplotypes is more methylated than the other. And we can then phase these also across the whole region. De novo assembly is something that we also have been trying, and this is a result for one of the samples where we ran the algorithm HiFi ASM on the HiFi data. A nice thing is that it took just 3.5 hours to do this analysis, and we got really good results, I would say. So we can span here 3.1 gigabases of sequence, and we put this genome together into just 373 contigs. You can also see the statistics here on the contig N50 length and so on, and it's looking very good, I would say. And when we then take these contigs, we can also map them back to the human reference genome. And here in the figure to the left, you can see the different contigs that are mapped to the genome, and they are colored in red and then blue, just to see sort of the changes between the different contigs. So it just shows where the breakpoints between the contigs are. And as you can see in this figure, we were actually able to put chromosomes 11 and 18 together in one single contig, which I think is quite amazing compared to what we have been able to do before. But there are also still white parts here in the genome, and those could be either repetitive regions or regions where the contigs fail to align uniquely. But the nice thing is that we now also have the T2T genome, and if we do the same exercise here, mapping the contigs back to T2T instead, we can see that we actually fill in some of the parts here that are marked in yellow with our contigs. So it seems like our de novo assembly covers more bases than the HG38 genome. And to really make use of this, maybe we should instead move to the T2T for some of the analysis that we are doing. But we're still sort of thinking about exactly how to do this. When looking at the causative SVs, because these were now patients that we sequenced, you can see one example here of a translocation breakpoint. And it's a very clear picture, I think, and shows exactly where the breakpoint is located. And you see the sort of rainbow regions. Those are the parts of the reads that are aligning to a different chromosome. So those are soft clipped reads. So we can in this way identify the breakpoints very exactly. But we're also looking at more informative ways to visualize these SVs. So one way would be through what we call subway plots, as you can see here to the left. And here you have a region on chromosome 3 with parts that are called A, B, C up to I. And below you can see how these regions fit together in the patient that we're studying. So fragment A is connected to C, and then to F, and then it goes to D, and to H, and then E, F, G, I, H, I. And the part B here is actually deleted. So this can be maybe a more informative way to sort of get the whole structure of this complex SV. And to the right, we see another way of looking at perhaps larger scale SVs and translocations. So here we have a translocation between chromosome 10 and 1. So there's a line going between these chromosomes. And also on chromosome 2, we have some kind of, I think, deletion of a part of the genome. So we're thinking that maybe these types of pictures can be easier for example, a clinician to look at to understand what's going on. Okay, so try to summarize these results. We obtained more than 20x HiFi coverage for all of these samples, despite that the DNA was suboptimal in some cases, or at least that there was not enough DNA to do a proper size selection. And when it comes to the results, we found about 5 million high confidence SNVs per sample, more than 20,000 SVs per samples. We got genotypes for most of the tandem repeats. We can phase haplotypes and also get the methylation profiles. And a very important thing is that 13 out of these 14 cases have now already been solved, and we have good hopes also for the last case. But one issue here is that sort of when we do this type of analysis, many of the variants that we find are novel, and the frequency of these variants in the population are basically unknown. And this means that sort of if we would do this on a patient where we have no idea where the causative variant is, it would be difficult to rank the different variants and predict the variant effects. So trying to a way to solve this would be to do a whole genome analysis for populations and sort of gather the variant frequencies in a big database. So I think that would be something that would be very nice to do at some point. We already did it before for short read data. So we have had a Swedish 1,000 genomes project called SWEGEN, where we previously generated then Illumina data for 1,000 individuals. So you can see here to the left a PCA plot basically of the Swedish individuals compared to some other countries in Europe. And what we also did before was to take 2 of these individuals and sequence them with long-read technology. So we took 1 male and 1 female, and also from different parts of this cluster. When we assembled this, so this was back in 2017, and also the sequencing was done on the old like RS2 instrument. So we sequenced a lot of smart cells. The assembly was performed using something called Falcon, and we also did error correction afterwards with a program called Quiver. The assembly time was about 3 weeks per genome back then, but the results were quite nice, I would say. So we got assembly sizes of about 3 gigabases and 11,000 contigs. But an interesting thing is to compare this now to what we got from the Revio system. First of all, the assembly time went down from 3 weeks to 3.5 hours. The assembly size stayed about the same. We got a bit larger genome size assembled now, but the number of contigs dropped from 11,000 to 373, and the largest contig up to 159 megabases, and the N50 really increased, so from 9 megabases up to 56. So trying to summarize this, I mean, even before the 2 Swedish genomes that we sequenced a long time ago, they gave us a lot of information about novel sequence that was not in HE38, and also improved structural variant calling. But if we're thinking of the Revio system, we can now do this on a new, different scale. So we can sequence 1 sample per smart cell, and we can start multiple smart cells a week. The analysis time is about 4 hours, and we would get much better results for each sample that we're running. So I think it's really interesting to try to sort of think about how we can do this in a good way and start creating population scale projects. Okay, so jumping to the final subject of today, I will talk about some work that we have been doing on sequencing of single cells. So we're trying to sequence the whole genome in a single cell using long-read technology. And why would you want to do this? And I think that there are many reasons. One thing is, of course, to study the genetic variation between cells. It can be important for tumor evolution, neurogenetics, trying to find out de novo mutation rates, recombination, and lots of interesting questions. And the one other thing is that we basically don't know yet how the genome in a single cell looks. So it can be quite a challenge, but also interesting thing to do. But there's a big problem here, and it's that long-read technologies requires a lot of DNA. So for normal PacBio HiFi sequencing, we typically require 5 micrograms of DNA, which is equivalent to the DNA from 750,000 cells. There is sort of ways to do this more low input, to go down to 1 microgram, but that's still 150,000 cells. There are also ultra-low input protocols that are based on PCR amplification, where you can go down to 5 nanograms of DNA, but that's still 1,000 cells that we have to study. So an interesting question is how to study this. I mean, 1 human cell contains only 6 picograms of DNA, so how to actually sequence this using long-read technology. And what we did was to do of course, some type of amplification, and we did it to something called droplet-based MDA. And this works in this way that we have a cell here starting to the left, and the DNA fragments in this cell is isolated in different droplets. And within each droplet, we perform an MDA reaction that amplifies the DNA that is present in the droplet. And the reason why we do it in droplets is that we don't want one single fragment to sort of take over the whole amplification. By doing it in droplet, we sort of limit this reaction. And after the amplification, we break the droplets, get the DNA from the droplets, and sequence it either using short read or long read technologies. And for this droplet generation, we used an instrument from a company called Samplix and the instrument called XDrop. And the experimental setup of this study was that we started from 2 different human T cells. So one T cell called A and one called B. These were in vitro expanded, so we got several cells that looked the same, both for A and B. And then we took cells from these clones and sequenced them, both with short read sequencing and also with PacBio HiFi sequencing. And I will not go into everything in this project, but just show you some of the results. And this is one example of a region in the genome that is difficult to sequence with short reads. So you can see here at the top we have a region where the reads are uniquely lining. But in the single cell data, both from the cell A and cell B, we can cover this region and also call variants across this whole region. Another interesting thing was that we could find mitochondrial heteroplasmy in one of the cells. So here we see PacBio data for cell A and B on the mitochondria, and at one position we have a SNP, but it's not present in all of the reads. So that means that this cell B contains mitochondria that has different genetic variants. And we have also sort of sequenced several cells from this clone and see the same pattern. So it's actually a true biological finding. Another example is an insertion here of a region where we can see at the bottom we have one single cell here from clone A, where we have an insertion of I think 700 and something bases. And we see the same size insertion also when sequencing this same individual with bulk sequencing using PacBio. But in Illumina, this variant is not really seen. So I think that this can be a good way to find these types of insertions and, for example, transposable elements and try to study those things in single cells. And to summarize some of the findings from this study, we obtained about 1 million SNVs per single cell and about 5,000 large insertions and deletions per single cell. There were also lots of inversions and duplications, but we believe that most of those are false, that are caused by like amplification artifacts. We were able to detect some somatic variation between the T cell A and B, so 28 somatic SNVs, but no somatic structural variants or tandem repeats. And finally, we're also able to assemble part of a single cell genome. So we obtained a genome size of 600 megabases from one of these T cells. And I think the conclusion of this is that, I mean, it's a pilot study and more of a proof of principle that this works. But I think that long read sequencing can improve also the variant detection on the single cell level. And if you're interested to read more, we have a publication that is just out on this. Finally, I would like to mention that we're doing other things as well. So targeted sequencing, you already heard a bit about that from our node, using the twist hybridization capture. We're interested in single cell transcriptomics using the single cell mass isoseq method. And one of my favorite subjects is CRISPR-Cas9 editing and trying to see what happens after you have been doing genome editing. And I believe that we really need long read technology to verify the outcomes when we have done these types of genetic modifications. And finally, here is a slide with acknowledgements of the people that have been involved in these various projects, from the Karolinska Institute, from Uppsala University, SciLifeLab, and also the different nodes at the Genomics Medicine Sweden. And thank you all for listening.